Good day. Welcome to today's soft training titled GCLP. That stands for Good Clinical Laboratory Practice. Before we continue, I would like to invite you to please visit our website and like us on Facebook. This is not to spam you, but simply to receive regular updates on our services and training. First, let me introduce myself. My name is Hannes van der Merwe and I work in clinical trials since 2003. I've covered role in analytical chemistry and various compliance, working with GCP, GLP, ISO 17025, ISO 15189, and GCLP. I wish to introduce you to the regulatory environment today, and hopefully after this you'll have a more understanding of how to balance these different regulations and make them applicable to your lab. Today's session will be focusing on GCLP. Simply put, how to balance GLP in a GCP environment. For many labs, this also includes balancing ISO 15189. GCLP addresses and governs how organizations and their personnel operate. It has specific requirements for the facilities, the equipment, trial materials, and the reagent. As most guidelines, it relies on the policies and SOPs to achieve its goal. As with GLP, there's a lot of focus on the planning, which includes subcontracting, trial materials, conduct of the work itself, reporting of results, quality control, quality assurance and auditing, storage and archiving. As it is based on GCP as well, there is a lot of focus on confidentiality and safety. Before we go any further, there is something you need to realize. Complying with a guideline or obtain accreditation does not have an end. It is a continuous process of improvement. Guidelines get updated, but also how we understand and implement them changes. This changing regulatory environment is governed by a couple of institutions, the ICH, the EU Directive, South African Guideline, OECD, ISO, FDA, and I want to include even SANAS and RQA. Strangely enough, GCP itself has got very little about the requirements for labs. Therefore, GCLP ensures that there are systems and procedures in place for samples from clinical trials. This ensures reliability of the quality and integrity of the data. It ensures that data between different labs are comparable. It allows auditors afterwards to reconstruct the study. It also allows the uniformity of the performance of functions across different laboratories. First, look at the definition of GCP. According to the ICH, GCP is the standard for the design, conduct, performance, monitoring, auditing, recording, analysis and reporting of clinical trials, and provides assurance that the data and reported results are credible and accurate, and the rights, integrity and confidentiality of trial subjects are protected. The important thing for me here is that you really look back at the origins of GCP. And if we look at that, we realize it's the international ethical and scientific quality standard that involved the participation of human trial subjects in clinical trials, and that it had its origin in the Declaration of Helsinki. There's two main focuses, and the one is the confidentiality of trial subjects and the safety of trial subjects. As I said before, it actually has got very little requirements on how a lab should analyze the results. The one thing that GCP does say is that it wants the samples to be analyzed according to the principles of GLP. Now, GLP itself is a managerial concept dealing with the planning, monitoring, and as well as the reporting and the recording of results. It is also specific for preclinical and non-clinical work. Although it is great to promote the quality and validity of data due to its documentation focus, it is kind of a mismatch and does not really satisfy all the needs we have for a clinical laboratory. So out of this need for guidance that's specific for clinical trial labs, GCLP was developed between the World Health Organization and Research Quality Association. To look at the definition is quite a mouthful. GCLP is applying those principles established under GLP for data generation used in regulatory submissions, which are relevant to the analysis of samples from clinical trials, while at the same time ensuring that the purpose and objective of GCP principles are satisfied. In doing so, it ensures that the reliability and integrity of data generated by analytical laboratories can be assured, kind of balancing the world between the safety and confidentiality of the GCP side, as well as the documentation integrity and the trustworthiness of results from the GLP side. Okay, so for any trial kind of following GCLP, there's a couple of definitions and role players we need to look at. One is that for the clinical side, there will still be the GCP regulation. So there will be a kind of documentation and trial management files that needs to be in place. Separate from that, GCLP then requires from the lab to have an analytical plan or analytical protocol, if you can call it that. There should be an analytical project manager or trial manager. In GLP, this would be referred to as a study director. For GCP, which we'll call that the principal investigator. But remember, that's now separate. There should be facility records in place that details with everything, all the equipment, access to the building, that kind of stuff. The investigator should be listed from the principal investigator for the clinical trial itself. So there's a cross-reference here. There should be systems in place for quality audits. That's obviously done by an independent party, but we'll look at that in more detail later. Raw data, all raw data for GCLP studies should be kept. And remember, raw data is that first place where you document something, or the first recording, or the first printout. The results should be obviously as Excel sheets or data sheets, or even can be summarized in an analytical report. The sponsor remains the same as for GCP trials, so that's the person with the interest of specific results, or kind of who's paying for the research to be conducted. Trial facility is where we're going to analyze the samples, in this case, not where the patients are kept. The trial facility management, this is the lab management, and this is normally the upper management of the lab, the lab directors, upper lab management. The trial material is the substance that we're going to analyze, the molecule of interest. The trial protocol, again, is like the analytical plan. This contains all the detail about when the study is going to start, who's going to be involved, and how you are going to report your results and according to what guidelines. So let's look at the organization and the personnel involved. Who is the trial facility management and what's their responsibilities? As mentioned, this is possibly the lab director, the unit heads or the managers involved. And they are responsible for a couple of things. They need to ensure that the personnel working on a specific clinical trial has received the training they required, and that they do have access to suitable equipment and facilities. If you look at the role of the analytical project manager, this is a very central and very important role. This person is normally responsible and the main contact between the trial facility management as well as the sponsor. It's a central point of communication. He or she would release the results to the client and ensure that the client's needs are reported to the lab staff and to management. Trial staff also has responsibilities, and that is to read your SOPs, to read the policy, to read the trial protocol and ensure that you comply with all the requirements thereof, as well as the requirements of regulation. Interestingly enough, a GCP guidance put the responsibility at personnel to know the regulation, and it's not necessarily the trial facility management to ensure that you know your regulation and comply with it. Remember, we are still here pre-analysis, so this is kind of everything that should be in place before we even take on a clinical trial or GCLP trial. So trial facilities, trial facility is where we're going to do the analysis. This is the labs, all the rooms and stuff that we need to do our work, from balance room right through to freezers and fridges. And they need to be of suitable size and construction and suitable for the job intended. We also need archive facilities. Now, I remember archive facility for clinical trials. We need to be able to store documents up to 14 years, and that needs to be kind of climate controlled and protected from moisture and protected from light and protected from insect. Luckily, this is one of the things that we can subcontract. And because the requirements for these are so specific, even with fire control and the temperature and monitoring and stuff, I would actually recommend most companies that want to get GCLP accreditation to rather subcontract this to a kind of already accredited facility. Then waste disposal. Now remember, we're not only dealing with medical waste disposal of samples or blood samples or serum samples, we're also dealing with possible chemicals that we need to get rid of, and GCLP simply requires that you do this according to local regulation. So it's not specific to tell you how to do it. It should be done the way that the laws of the country that you're operating in is doing it. When we look at the equipment, the materials and the reagents, the following is listed as a requirement. Equipment for any kind of clinical work should be suitable. It should be well maintained and operated by qualified staff. It's not only a simple case of kind of deeming something to be suitable and well maintained. You need to have the documentation. So if you're going to say that a piece of equipment is suitable, I would like to see the installation qualification. I would like to see the performance qualification and the operation qualification of that specific piece of equipment if I was the auditor looking into this trial. And then obviously also the maintenance log and the training logs of the staff involved. So materials, they should be appropriate and well controlled. So I can't simply say that I received a batch and I used it. I want to know what date was the specific bottle of reagent received. I want to know when was it opened, and I want to know when is the expiry date. So someone should maintain logs for all the stuff that get ordered, how they get received, and when I need to get rid of them, when they need to be replaced, and basically like stock control. So reagents, so this material that I order, I'm going to make different buffer solutions or acids with them, and whenever I make these, I need to indicate clearly what it is. So all bottles and reagent bottles in the lab should be clearly identified. It should have a preparation date, it should have a storage temperature, it should have an expiry date on there, as well as the person who prepared it. SOPs is needed for all clinical trials as well as GCLP studies. We need an approved set of SOPs and policy to be in place. They should be signed off by the appropriate staff and should be made available to everyone involved. There should be SOPs for all activities. SOPs form a critical part of any quality control system. Only if I've got all these things in place, the suitable personnel, suitable equipment, suitable facility where I can conduct my work, only then I can start planning on my GCLP study. So I need an analytical plan. That analytical plan needs to detail how I'm going to receive the samples, how I'm going to generate results, who's going to be involved, possibly the name of the analysts, contact details of who should be informed about anything funny that happens during a clinical or during the analysis. You should remember that subcontracting for this kind of trials is allowed, but subcontracting can only be done with the permission of the sponsor, and therefore needs to be included in the analytical plan. There should also be systems and processes in place for the trial material. Now remember, the trial material is that compound that I'm going to test or screen for or try and monitor in my various subjects. And those I'm going to receive, for those I'm going to receive some reference material from my sponsor. And I need to have proper systems in place to track and monitor where I received them from, how they were stored, and also how they were used. So if I receive, for instance, 20 milligram of the reference material, at the end of the trial, the sponsor can require from me to indicate how much I have left, and I've got 10 milligram left. I need to indicate what happened to that 10 milligram that went missing and how it was used. Okay, great. So we've got the facility, we've got the staff, we've got the equipment, we've done proper planning, everything is documented, everything is signed off, and now we can start doing our work. Now we need to remember, I can only do what I've planned in my protocol and in my study phase plan, and which is allowed according to my SOPs. And I can only do this on computer systems that's been validated, using methods that's been appropriately validated. Before I even kind of decide to repeat some of my results, I also need to kind of have systems in place for how I'm going to repeat some of my analysis. If anything is found to be not in line with what I expect, I cannot simply decide, oh, I'm going to redo the sample. There need to be a planned system already in that protocol that we refer to. And if we didn't have a plan in place for how to identify samples to be repeated, I'm possibly going to have to stop my clinical trial, get permission from the sponsor, and issue a new protocol or plan, and only then I can continue and identify those repeats and continue with the rest of the clinical trial. Then after my study has been completed and all the review process has taken place, I can either release the results as data files, or I can upload it on the internet on an agreed shared server with the specific client, or it can possibly be written in an analytical report. Now, an analytical report is a lot more work than simply issuing the results as a log or as an Excel sheet. So this needs to be agreed with the client before you start the whole clinical trial. It should be included in your study phase plan or in the trial protocol. One of the things I really appreciate if you kind of take home from this training session is that there's a big difference between quality control and quality assurance. Quality control can be many things. It can be a step, it can be a person, or it can even be a sample. So its intent is to verify accuracy. It can be simply asking your colleague to verify your results. It can be the inclusion of a sample to verify the accuracy of your calibration line, or it can be the membership of an external performance proficiency scheme where another lab or a facility sends you samples on a regular basis. You analyze these and then prove that your system's quality is still in check. Quality assurance is something totally different, although part of it is to check that a QC process is in place. Quality assurance is ensuring compliance with the trial protocol, the analytical plan, the SOPs, guidelines. It also includes the facility, system, equipment, method, QC processes, personnel, and documentation in its audits. These people are normally independent. they do not report to the same manager than the lab staff would. They should be competent, they should record their results, and they directly report to upper management. After an audit, they would normally issue a certificate. It is very important to realize that they are independent, and what they normally do is that they will do a very small QC check on the data as well, where QC process normally does 100% QC check. Quality assurance has got a process of selecting a small part of the data. Now remember that we need to store documents after a clinical trial has been completed, and the regulatory authorities normally want the essential documents for clinical trials to be stored at least 15 years. But there's a lot more that can be stored, things like personnel files, SOPs, the data, samples itself. And if you did not agree to something in the protocol, this can be a very costly exercise. Make sure that before the study starts, you've got the agreement in your study protocol in place with your specific sponsor, how long you're going to analyze, still store samples for future analysis, or whether you can discard it after a specific time. And please, you cannot simply discard these. You need to have a documented destruction certificate in place. So when you send this for collection by your disposal company, normally you can indicate on the forms what they are destroying, and they will be able to issue a small certificate and keep this on file that you can in future for orders make this available. Now remember, GCP is very strict on confidentiality. So if we look at the confidentiality of a GCLP trial, it's basically got the same requirements. So you need to make sure that you do not divulge names of participants in the clinical trial. Confidentiality is also governed in the protocol and as specified in the Declaration of Helsinki. Most of all, the confidentiality of the results is according to the requirements of the sponsor. So even releasing results to the sponsor, you need to ensure that you understand the way that you need to release it, whether the data is blinded, whether it is double blinding, and if you release results to them and it is not in the right manner, you can compromise the outcome of the trial. So also, before you even start the clinical trial, make sure that there's a section on confidentiality and releasing of results in your clinical trial protocol. Further looking at the ICH GCP, it is very clear that the responsibility of ensuring there's an agreement on the confidentiality of the data is actually the sponsors. However, there's no harm in asking. Before the trial starts, it's better just to ask than to say, tell the sponsor afterwards it's their fault and they should have asked. I mean, it's just for good relations, just ensure that something in place. As mentioned, GCLP has got a strong focus on documentation. It's based on GLP and GCP. Again, GCP being the confidentiality and safety side, origin in the Declaration of Helsinki, with GLP is to ensure reliability of data, and therefore documentation, planning and doing according to plan, and making sure that everything is documented. It is the show, not the tell, and documentation is the show. A PDF of this training will also be made available and emailed to everyone attending. And there is just some very handy references if you want to do some more reading. GCLP on its own is actually a training that should go over a couple of days with practical examples, and also detailing a lot more on the sections like SOPs as well as GCP. I would like to invite you in the future to attend sessions that I'll present on SOPs and GCP itself. Okay, this brings us to the end of a very brief overview of GCLP. There's a lot more to it, obviously. We can have a lot more discussion on SOPs, and there's actually a lot more that we should discuss on GCP itself. SAF regularly brings out the more sessions, and we would also like to tell you more about the services, so please follow us on Facebook and visit our website, and we'll keep you updated on any future sessions that will include SOPs and GCP. For any further questions or discussions, please contact me on my email, and hopefully I can explain more clearly anything that you didn't understand. Have a lovely day.